All right, we'll get started. Thank you everybody for joining. I'm going to start with some brief housekeeping. Just first to check, sound check, Lukasz, if you can hear me well. Thumbs up, perfect. All right. So hello everyone. My name is Christoph Diaz. I'm the Vice President, Scientific Affairs at curePSP. Welcome to our second Ask the Scientist monthly webinar. We received a lot of encouraging feedback from last month's presentation, and I'm pleased to see that our registration number have almost doubled today. Before we get started, a few points I'd like to make. A reminder that you are automatically muted and that your video is disabled as you join the webinar platform. During Dr. Joachimiak's presentation, please use the Q&A function to type your question, and we will do our best to address them at the end of the 40-minute presentation. The questions that will not be answered due to lack of time will be addressed and shared to registrants via follow-up email early next week. Our speaker today is Dr. Lukasz Joachimiak, an assistant professor at the Center of Alzheimer's and Neurodegenerative Disease at the University of Texas Southwestern Medical Center. And he's also a recent curePSP Venture Grant awardee. During his Ph.D. at the University of Washington and his postdoc at Stanford University, he developed computational approaches to understand the relationship between protein conformation and protein function. Today, he will tell us how he's applying his methods and approach to better understand tau, its structures, and its ability to form aggregates. Before we get started with the presentation, on behalf of curePSP, I'd like to thank our thousands of donors, volunteers, and supporters. All that we do at curePSP, including this new webinar series and the research we fund, is made possible by the hundreds of donations we receive each month. So thank you all for your continued support, especially in these difficult times. Dr. Joachimiak, thank you for sharing your time and expertise with us today. If you want to share your screen, we can get started. Thank you so much.
Can you see my screen?
I can see your screen.
Excellent. Thank you for the wonderful introduction. I'm excited to share my lab's work to everybody today, and it's actually sort of nice to not have to travel for a talk. So I get to give it from my, the comforts of my office. So today I want to tell you about my lab's work on the structure of tau and how it changes conformation to adopt different fibrillar conformations that now we sort of have insight from Cryo-EM structures. Okay, so this audience probably doesn't need a reminder that the formation of amyloid fibrils is associated with the formation or with disease in human brain. And so here is a set of different proteins that are all associated with the deposition of amyloids in the human brain and disease. And so we have alpha-synuclein, this is associated with Parkinson's, MSA, et cetera. We have amyloid beta and tau are linked with Alzheimer's disease. The Huntington protein is associated with Huntington's disease. TDP is associated with ALS as well as FTD. And then PRP is associated with Creutzfeldt-Jakob disease. But the main thing I want to sort of highlight is that we maybe know the conformations of these fibrillar states associated with the fibrils, but we know very little about the conformations that drive the formation of these structures in the monomer. And so tau is actually a very exciting sort of example because the monomeric form of tau is actually extremely soluble. Tau you can purify and you can boil it and nothing will really happen. It won't aggregate. In fact, you can let these samples sit for quite a long time and you won't see fibrillar conformations. And so really it's unclear how this aggregation resistant state changes conformation to drive to the formation of these amyloids that are the hallmark of disease, for example, Alzheimer's disease. But in fact, it's actually more complicated because this one tau protein sequence actually has the capacity to adopt a diversity of different stable fibrillar conformations that are all linked to disease. So Alzheimer's disease, Pick's disease, PSP, CBD, really the list goes on. There's over 20 tauopathies that are linked with tau fibril deposition. And we really don't know how this soluble aggregation resistance state changes conformation to these different states. And so with recent cryostructures that have been published from the Scherer's group and also from the Fitzpatrick group who came from the Scherer's group, here are structures of tau fibrils that are isolated either from patient material or from recombinant sources induced by heparin. And so here we're looking at the down the fibril axis of each structure. So here we have two conformations isolated from Alzheimer's disease. And so you can see that the monomer conforms this sort of C-shaped conformation. And that conformation is identical between these two different fibril types, PHS, paired helical filaments, and straight filaments. But then the way that they interact to form the fibril is actually quite different. In Pick's disease, we have a very different conformation. This is actually associated with the specific tau isoform 3R. We also have a structure of CTE. This is associated with brain damage. And of course, we also have the more recent structures from CBD, which have a yet different conformation from these other structures. And then of course, the heparin structures are also quite different. So really from one sequence, we get the diversity of different fibrillar conformations, and a subset of these are all linked to disease. Okay, so the tau protein that's predominantly present in the brain is this isoform that's 441 residues. It contains these 4 repeat domains that are essential for binding to microtubules. And in fact, this is how tau was originally discovered. So it co-purified with microtubules, and this is discovered by Mark Kirschner when he was at UCSF. Over the course of the last 30 years, we've also learned that tau can encode mutations, and these mutations cause inherently inherited disease that have 100% penetrance. And what I want to highlight is that the mutations, for the most part, localize to this repeat domain. And sort of for the purpose of this talk, I want to also emphasize that the mutations not only localize to these repeats, but they also localize to these interfaces between the repeats here. And now, additionally, what we know about tau is that it encodes these little motifs. We call them amyloid motifs. And these motifs are essential for the driving of tau to form aggregates. And so this sequence here, VQIVYK, is one of the predominant sequences, but then there's at least 2 other sequences that drive amyloids in tau. And so if we color code now the structures I showed you on the previous slide according to these different repeats, what we find is that not only can tau adopt different conformations in these fibrillar states, but they use different elements to stabilize these structures. And so how does this aggregation-resistant conformation change shape to drive the formation of a diversity of different structures that are associated with different diseases? And so about 2 years ago, in collaboration with the Diamond Lab, we identified an alternate conformation of a tau monomer that is actually aggregation prone. And so for the rest of the talk, I'll refer to this conformation as the seeding monomer, or MS. And then the conformation that's aggregation resistant, I'll refer to as MI. And so for the purposes of this talk, I want to sort of introduce to you some of the methods that we use to study protein aggregation, both in vitro and in cells. So in vitro, we can follow tau assembly using a dye called thiophosphatase. This gives you a fluorescence signal as tau assembles. And so you can watch the protein assemble as a function of concentration. We can also follow the actual size, the radius of hydration size of the molecules using dynamic light scattering. And then we can also directly image the fibrils using transmission electron microscopy. In addition to this, we can follow aggregation in cells. And so this is an assay that's developed by the Diamond Lab. It's called the Tau biosensors. And it really harbors, it's a mammalian cell that harbors tau fused to fluorescent proteins, so CFP and YFP. And if you transduce fibrils or seeds from the outside into these cells, this induces aggregation of these proteins, aligning them, and now your YFP and CFP proteins are next to each other. And this allows you to detect the formation of these structures using FRET fluorescence. Okay, so the first experiment that we did to try to isolate the species in disease is to use immunoprecipitation coupled with size occlusion chromatography, which allows us to separate the proteins by different size using human brains. So we used data or samples from AD-derived patients and also age-matched controls. And then we assayed the different size fractions using the seeding assay that I just showed you. And so on this plot, we have the different size molecules on the bottom. And the y-axis corresponds to the extent of seeding capacity of the different samples. And so our age-matched control did not yield any seeding across these different conditions, or fractions. But in contrast, the AD sample yielded seeding for large things that were greater than 10 molecules, 10 molecules, 3, 2, as well as 1. And so the observation that a monomer gave us seeding was actually quite surprising and controversial. And so this sample, this monomeric seeding conformation of tau derived from AD has interesting properties. It has the capacity to self-assemble. So if you purify this sample and you let it sit overnight, it'll re-equilibrate into these larger species. And it can also introduce or induce aggregation of naive conformations of tau to get them to aggregate as well. And so we wanted to ask, in terms of appearance, when do these species first occur in disease? And so we turned to a mouse model to try to study this. And so we used the PS19 tauopathy mouse model, and we followed the same procedure. We can use an IP to purify tau, and we can separate them by different sizes, and then we can measure seeding activity across these samples. And so in terms of this mouse model, you really don't see pathology or the formation of insoluble species until about 6 months of age. So the observation of certain pathology is really sort of later. And so what we found is that in this mouse model, we don't really see seeding for soluble species until 4 weeks. So at 4 weeks of age, we see seeding, but this is not from insoluble species. Okay, so let me orient you this. So here on the y-axis, we have the extent of seeding. This is on a log scale. And then we have data for weeks 1 through week 6, and then 1 year when we have pathology. And so SI is insoluble species, SS is soluble species, and then we have the different size fractions. So for the first 3 weeks, we really don't have any signal. At 4 weeks, there's no signal for insoluble species, but we see soluble signal, including monomer and larger species, but predominantly small species. At 5 weeks, the soluble species increase, insoluble is still flat, and then that maintains at six weeks. And then at 1 year, of course, we have insoluble species and we have the distribution of the different soluble species. So this really suggests that the seeding monomer really is the incipient species that drives disease, okay? And so we wanted to ask, what are the properties of this species? People have proposed that phosphorylation may be a driver of this. And so we wanted to ask if phosphorylation is obviously different between these different forms of seeding monomer in a mouse model, and then we also compared AD samples. And so we did the same experiment, but now we use mass spectrometry to identify sites of phosphorylation across these different seeding monomer species in the mouse model. And so this is color-coded by the different peptides. The peptides that have no modification are colored in cyan, and then peptides that have heavy phosphorylation are colored in magenta. And so what we see is that there's maybe a bit of a difference at the N-terminus for weeks 1 and 2. This is not a dramatic difference. But then 3, 4, 5, and 6 are really essentially identical. So we don't have a transition between weeks 3 and 4 that would be suggestive of a dramatic change in phosphorylation patterns that would drive disease. We did similar experiments on samples from age-matched controls and AD. And again, we don't see dramatic difference in phosphorylation. And in contrast, the AD fibril does have different patterns. And so in the monomeric state, there doesn't seem to be an obvious modification that seems to drive this process. So to validate this observation, a student in my lab, Dailu Chen, developed an assay where we can purify these species and we can treat them with a phosphatase, confirm that the phosphatase remove the phosphorylation pattern, and then assay whether we have changes in seeding capacity. So we can measure the change in phosphorylation after phosphatase treatment using a phosphate gel. So we can see the shift from this band down to this band, so we can collapse this into a single band. This is also true for the AD samples. By STS page, they look about the same, or Western. And so now we can ask if the seeding capacity changes when we remove the phosphorylation. And so in gray bars here is the mouse data. So this is 3 independent biological replicates. And so without phosphorylation or without phosphatase treatment, we have some seeding. And if we treat the samples with phosphatase, the seeding really doesn't change much. So phosphorylation doesn't seem to be a driver for the capacity of these species to seed. And of course, we see similar data for the AD, although there's some more heterogeneity. For this one specific sample, we had an increase in seeding. Okay, so we can try to purify these species also in vitro. And so in vitro, you can produce this sample of tau fibrils by incubating tau with a polyanion heparin. If you mix tau with heparin, and over time you produce fibrils. And so we wanted to ask, can we produce these species in vitro and then trap them to capture intermediate species. And so this is exactly what we did. Again, we're using size exclusion chromatography to separate the different species. And so here is our inert tau, the tau that does not aggregate. We have this nice peak that elutes at about 13.5 mils. If we incubate tau with heparin, what we find is that the peak elutes earlier and yields an oligomeric sort of peak right here, okay? One of the obvious questions is the heparin co-eluting with tau? And this is why we get a bigger species. And so this is why we used heparin that has a dye on it. And so we can see that free heparin with the dye comes out right here. And then if you mix tau with this heparin, the peak does not change elution profile or shape. And this is very sensitive. And so we don't really see any signal for the dye in the sample. We've also used other biophysical approaches to characterize the molecular weight of these samples. And one of the limitations of these methods is that they require high concentrations of protein. So at about 2 mg per mil, if we do this experiment, we see that the sample here is actually a mixture of monomer and dimer, but there's ways for us to control whether it converts into this. But we actually had access to a sort of a new instrument that allows us to measure the molecular weight using scattering interferometry. And so this is it's a mass photometry experiment. And so the MI conformation here is clearly a monomer at 50 kilodalton. So this matches the size perfectly. And at this concentration, the seeding monomer is predominantly in the monomeric state, and then we have some small fraction of dimer. And if we titrate the concentration, we can see this redistribute to the monomer and dimer. And if you go to lower, you see mostly monomer. And so we really have confidence that the experiments that we show seeding, we actually are probing the conformation or the capacity and activity of monomer to induce aggregation. Okay, so now we have this interesting new species. This species has the capacity to self-assemble, and so it's sort of intractable to classical structural biology approaches. So NMR has sort of been one of the main methods that people have used to study the structure of tau. But of course, these are long experiments. They typically require high concentrations of protein, so it's not trivial to probe the structure of the seeding form using NMR because it would simply assemble. And so I want to tell you briefly about a method that allows us to interrogate the properties of the sample first, and then we'll talk about the structure. And so we can transduce the seed into cells, and we get aggregation. Our non-seeding form does not aggregate. We can also induce aggregation of tau by spiking in small amounts of the seed. So we use 4 micromolar tau, and then we add small amounts of the seed to induce aggregation. We can follow the assembly using Ph.D. or DLS. And one of the remarkable things is if we add tau and heparin and we follow aggregation, we have this long lag. If we add these seeds, even at substoichiometric concentrations, we don't have a lag. So aggregation is nearly instant. And it's really concentration dependent on the amount of seeds. So if you add more seed, you get more aggregation. And if we follow the samples at the very end, we can see that by TM we form fibrils, whereas a normal tau does not, okay? And so I want to tell you briefly about this method that we've developed to understand protein structure. Requiring small amounts of protein, we can probe samples that are isolated from tissues, et cetera. And so this has been a very powerful approach that allows us to interrogate sort of large complexes and dynamics. And so we wanted to ask whether we can use this approach to study more intrinsically disordered proteins that don't really have strong structure. And so this method combines cross-linking with mass spectrometry. So we can take a protein that has some structure, some contacts, and we can use a chemical cross-linker to trap an interaction between 2 different elements. And then we can digest this into smaller fragments and then use mass spectrometry to identify the contacts between these. And so this way we can infer the structural information from these different points of contact. And so this is exactly what we did. We can take our 2 different forms of tau, react them with a cross-linker. We can confirm that the samples are not forming large species under the conditions that we're probing the structure. And this is important because what we're interested in is understanding the intramolecular contacts in the monomer and not intermolecular contacts. And so if we take our MI conformation, react with cross-linker, it remains monomeric. If we take our MS conformation at these concentrations, even after 10 minutes, we don't see an appreciable formation of large species. Okay, so what does the data look like? So here is the data for the aggregation resistant state, MI. And so each loop here is a contact point between 2 positions in the sequence that we identify with the cross-linking method. So MI gives us this sort of mixture of long-range contacts, as well as these short-range contacts. But it's really sort of heterogeneous, and this is indicative of the protein being dynamic. In contrast, the seeding conformation of tau had this sort of bimodal distribution of contacts. So you have contacts from about 150 to the first or second repeat domain, and then P1, P2 here to the C terminus. Okay, so the patterns of these two molecules were quite different. In the process, the review process, we had also shown that the seeds were actually resistant to heating. And so if you heat the samples over time, they still retain some seeding. And so we wanted to ask what happens to the patterns of crosslinks if we heat the samples. And so if we heat the normal aggregation resistant state, it fully unfolds. We don't really see crosslinks. But in contrast, if you take the seeding conformation, we preserve a subset of contacts that we think are essential for seeding capacity. Okay, so now we have information about the different sort of topological contacts between these two different forms. How can we translate this into structural information? And so we can use these crosslinks sort of as guides, as NOEs, for example, in an NMR experiment. And so these are long distance sort of contacts that can be used as geometric constraints in simulations. And so coming from the Baker lab, we can actually employ these in simulations using Rosetta to build an ensemble of structures that are consistent with these geometric constraints. So let me show you what this data looks like. So here are ensembles of structures produced for the MI conformation, driven by the experimental restraints. And on the bottom are the ensembles of structures produced for the MS. Each point is a different structure, and it's separated along an axis of energetics versus the size or the radius of gyration. And so the sort of obvious thing that came out of this is that there's a slight change or perturbation of the average size of the MS versus the MI. MS appears to be a little bit more expanded than MI. And if you look at more closely at the features that distinguish these two structures or ensembles, what we found is that in the aggregation resistant states, tau adopts these local structural beta hairpin elements surrounding these amyloid motifs that are essential for aggregation. So really this posed a very interesting hypothesis that in the aggregation resistant states, the amyloid motifs are actually buried and not solvent exposed to allow further aggregation. In contrast, what we found is that for the seeding conformation, these hairpins were predominantly extended. So we have a very simple hypothesis. The solvent exposure of the amyloid motifs is what determines this difference between being aggregation resistant and aggregation prone. And what's further more interesting is that pathogenic mutations that are linked to disease all localized to these elements where we have these conformational changes. So P301L is a mutation that's linked to frontotemporal dementia. This is a very aggressive mutation. And we see that it falls within this local structure, and we would predict that changes in sequence here could drive the formation of different structures that would expose the amyloid motifs. So how do mutations play into this whole process of tau aggregation? So there's been lots of NMR studies on tau with mutations, but nobody's really sort of framed structurally what happens to tau that drives aggregation. And so we asked first, this has sort of been known, but we wanted to do this ourselves, does introduction of mutations drive aggregation of tau? And so here we have data for wild type tau, and this is induced with heparin, compared to P301L induced with heparin, and we can see that there's a shorter lag for the mutant compared to the wild type. So the wild type is slower to aggregate than the mutant. We can assay these samples in our FRET assay and ask if these endpoints also induce aggregation. And indeed, all our controls behaved, so we can transduce fibrils, make sure that everything works. And we see more aggregation for the mutant compared to the wild type. And even after 5 days, we can see that the mutant gives you some signal compared to the wild type. Okay, so the mutant appears to be more aggregation prone. But what does this actually mean in terms of the structure? How is the mutant changed in terms of structure compared to the wild type? And so we performed sort of a nifty cross-linking experiment where we can couple changes in temperature to follow unfolding using cross-linking mass spec. And so we compared the wild type protein to the mutant P301L and then asked how are the changes in structure responding to temperature. So this is what the data looks like. So each of these points is a cross-link between this position here and this position here. and they're color-coded according to the frequency, how often we see them across a number of experiments. And I've broken down sort of the regions of contacts as local N-terminal, local C-terminal, N to C, and then also things that are within where the mutation is, okay? So we can summarize the data as a bar plot, counting the number of crosslinks in these different regions. So for the wild type, what we see is that the N to C contacts drop off. The N-terminus is fairly stable. The C-terminus is very stable, but then we also drop off the signal for the crosslinks where we have this proline at position 301. In contrast, if we do the same experiment with our mutant, our long-range contacts from N to C drop off almost to 0. We almost drop off to 0 for the N-terminus, and then we also lose more dramatically the contacts close to the mutation. So really, we conclude that tau is more susceptible to unfolding when it has these pathogenic mutations. And we also predict that there's local conformational changes in proximity to the mutation as highlighted by the loss of contacts here. Okay, so we can also run simulations without any restraints, no crosslinks, and then ask, how often do you form these hairpin conformations? So here is an ensemble of structures for tau using chemical shifts from the NMR experiments. And again, each point is a different conformation. And so we get this distribution of shapes as a function of energy. And if we just take this structure randomly, we see that this R2 element that contains the biquivic motif adopts a hairpin. And we have this PGG motif. Here is where we saw this mutation that changes conformation. And we see in the whole ensemble, about 60% of the structures have something that adopts this collapsed conformation. So this really gave us the idea that a minimal sort of hairpin may be sufficient to capture the local conformational changes. So this is exactly what we wanted to test. And we wanted to test this in the context of all the other mutations that are localized within this region. So here is this hairpin again, and we can see that there's 5, at least, sites that are mutated in the context of disease. And for the most part, these are all FTD-linked mutations. And so we wanted to compare the wild type sequence of this peptide in comparison to all the different mutants. And so again, we can assay aggregation using thioflavin T fluorescence. And so what we find is that the wild type sequence does not aggregate over the course of 100 hours. Okay, so the wild type element is actually aggregation resistant, just like tau. But in contrast, P to L and P to S substitutions aggregate within hours. And then all the other mutants have a spectrum. We can confirm that these all form fibrils. We can confirm that the fibrils transduce into cells. And so these are all functional aggregates. And so we wanted to ask, can we produce conformations of tau or design sequences that are actually now aggregation resistant? We have a very simple model where we have a hairpin, and if you add a mutation, we think the hairpin changes conformation, exposes the amyloid motif, and therefore drives aggregation. And so we wanted to install or engineer a sequence by introducing a tryptophan zipper at the end of the sequence. And so if we add this tryptophan zipper at the end of both the N and C termini of our peptide, we anticipate that the interaction would overcome the opening of the hairpin and prevent aggregation. We also created these controls where we have singles, and this is what we expect the model to look like. So we have a hairpin, we have a P to L substitution at this sequence here, and then by installing this sort of short linker with the tryptophan on each side, we expect that the indole interactions will overcome the reopening of the hairpin due to the mutation. And so indeed, what we find is that the P to L aggregates as expected very quickly. The left and the right sides also aggregate, but now our double tryptophan zipper construct completely abolishes aggregation. We also created biosensors to test this idea, and biosensors containing these tryptophan zipper contacts really attenuate aggregation relative to the P301 by itself. So in vivo, we can, or in cells, we can at least validate that this conformational change can be controlled by additional contacts to prevent reopening and therefore aggregation. Okay, so I want to bring you back to this idea that tau actually has multiple of these motifs. So we've been focusing on this motif here between the R2 and the R3 elements. And these also occur across the other inter-repeat elements. So here is R2, R3. We have this conserved PGG beta hairpin motif followed by an amyloid motif. And really, we predict that these interactions between these ends are stabilizing. But these elements also exist in the R1, R2, R3, R4, and then also the putative R4, R prime. Okay, so the combination of these different elements protect tau from assembly, and then perturbation of these elements and the combinatorics of the perturbation of these elements can drive the formation of different structures. And so we can model this. So I had showed you before that the 2, 3 forms a hairpin. We can see that the 1, 2, 3, 4, and R prime also form hairpins to different degrees. So the R1, R2, we see that about 30% of the population forms fibrils or forms hairpins. The R3, R4 forms about 50% of the time, and the R4, R prime forms about 32% of the time. Okay, so we have a distribution of the different stabilities, and this suggests that the stabilities may actually impact the frequency of how often these elements are exposed to give you different contexts that can drive the formation of different structures. Okay, so inspired by some experiments from Tim Miller's group, who had shown that 4R tau appears to be more toxic in animal models, and changing the splicing form, so reducing 4R to produce more 3R, is actually protective. And people have published this in vitro experiments where 3R tau appears to be less aggregation prone than 4R tau. And so we wanted to minimize this system and understand the local elements around this 2, 3 in the 4R form and compare it to the 1, 3 element here in the 3R form. And they both include this Viquivic sequence, but the local sequence around it is different. So we really hypothesized that the local sequence changes around the Viquivic motif change the property of 4R compared to 3R. And so just to highlight, here is the aggregation resistance, 3R states. Here's a 4R state. There's really only 4 substitutions between the two. If you look at the wild type sequences, 3R wild type does not aggregate. So this sequence does not aggregate. Wild type also does not. But in install mutations where we mutate the proline to a leucine, the 4R form, the R2, R3, aggregates rapidly, as shown before. But in contrast, the 3R form that only has 4 substitutions does not over the course of 100 hours. So how did these 4 different residues change the properties of this peptide to change aggregation so dramatically? Okay, so we did a very systematic walk from one sequence to the other, where we can ask what single, double, and triple substitutions walking from this sequence to this sequence change the properties of aggregation. And so we can follow the aggregation as a t-half max. So this is the amount of time it takes to get the half maximal amplitude of our fluorescence signal. And so our aggregation resistant form here does not aggregate within 100 hours, so it's flat. So single substitutions don't really have an effect except for this one here. Doubles have this nice sort of drop. And so what's really cool is that we can identify pairs of positions that have a dramatic effect on aggregation. And it's these 2 positions that flank the hairpin, the stabilizing PGG turn. And so we think that these specific contexts here are really important for controlling the aggregation of tau. And so a student in my lab performed molecular dynamics simulations to understand the conformational sort of ensembles of one peptide versus the other to sort of ask what are the different interactions and is this consistent with what we see with our aggregation experiment. And we're actually in the process of actually doing NMR experiments on these peptides to sort of validate these molecular dynamics simulations. So we ran 5 independent simulations, and then we can look at the contact sort of plots. So we can ask how often the different residues are in proximity to each other. So this is what the data looks like. So here is our self-diagonal. So these are self-interactions, so this is expected. But what we're looking for is if we have a beta turn, you'll populate this cross-diagonal where residues from the beginning of the sequence are close to residues at the end of the sequence. And so what we see is that the R1, R3, which is aggregation resistant, forms this robust signal across 5 independent MD simulations. And so now we can look at these in more detail. So here's the average structure from the 5 simulations. They have a fairly low RMSD, and they all appear to be collapsed. The 4R form has similar features. It maybe appears to be a little bit more expanded relative to the wild type R1, R3. But if we introduce mutations, now we see a perturbation. So R1, R3, P to S, still has a hairpin. It looks sort of different from this, but this still doesn't aggregate. But in contrast, the R2, R3, which aggregates rapidly, now has a dramatic jump in the RMSD. And so the P to S substitution changes the flexibility relative to the wild type, but it's not as dramatic when you install this mutation in the context of the R2, R3. And so let me show you sort of how these interactions change between these two sort of endpoints. And so we can look at the aggregation resistance state. So here is the amyloid motif in blue. And so we can see that this conformation samples this hairpin quite often. And one of the obvious things is that it forms a sort of set of symmetric contacts that really stabilize the type of hairpin that we expect to bury the amyloid motif more efficiently. In contrast, the more aggregation-prone conformation still samples the hairpin, but less frequently and is a lot more asymmetric. And so what this means is that the amyloid motif is on average more exposed in comparison to the aggregation-resistant conformation. Okay, so I'm gonna come back to this idea that we think that the local structures around these motifs play a central role in resisting aggregation, and then that the perturbation of these structures drives the formation of different fibrils. And so we wanted to ask what happens to these amyloid motifs if we take a closer look at the fibril structures. And so we can take a look at the AD, the CTE, the Pick's disease, and the CBD structures, and then ask what are the amyloid motifs doing in these structures. And so what we find is that there's common themes. So for example, the Viquivic motif, that's essential for aggregation, is engaged in this hydrophobic contact in both the AD and the CTE structures. In Pick's disease, the Viquivic now does something else. It interacts with another amyloid motif, Quebec. And it's interesting because this interaction is absolutely conserved between the Pick's disease structure and the CBD structure. And then finally, in CBD, this Viquink motif does something similar to what Viquivic does in AD and CTE. It interacts with FTLK. So there's sort of a modular set of interactions that can be swapped between the different structures to give you the diversity of different conformations. So we wanted to ask, are these motifs actually important for the stability of the fibril? So a student in my lab developed a protocol where we can in silico mutate each residue in the fibril and then ask whether that residue contributes to the stability of the fibril. And we can calculate essentially predicted binding energy or delta binding energy. Okay, and so here what I'm mapping is in red are residues that when they're mutated to alanine have a dramatic change in the stability. So these are residues that are really important for the stability of the fibril. And if you look at these different structures here, AD, CT, Pick's disease, and CBD, we see that all the residues that are important in the fibril are on the inside. So they make interactions within the fibril, okay? And if we look at the different motifs, again, we see this Viquivic motif interaction that's conserved across AD and CT is energetically really important in all of these different structures. The Viquivic-Quebec interaction is also conserved energetically in the Pick's disease structure and the CBD structure. And the Viquink-FTLK interaction is also conserved. And then additionally, the other contexts we have are all nonpolar. So the residues that really determine the stability of the fibrils are all nonpolar contacts engaged in the fibril, within the fibril. Okay, so now I want to show you some really cool in vivo data. This is a project that's developed by a student in Mark's lab, really leveraging this idea of a biosensor, but sort of flipping on its head and asking, can you probe what interactions are important for the incorporation of tau into different types of seeds? Okay, so we can make a library of mutants where we can substitute every single position to an alanine. We can make an arrayed lentivirus library of these, and then we can transduce these into a sort of strain. And then we can ask if the mutant can incorporate into that strain. And so we can read this out using FRET, using flow cytometry. And so things that have high incorporation are substitutions that are not important for incorporation. Whereas if you have low incorporation, these are alanine substitutions that do not allow incorporation into these structures, okay? And so we can compare this across a panel of different strains, and I want to just show you one sort of specific example of how this relates to structure. So we can take a CBD-derived strain, and then we can build this profile and compare them across different things. So here's a comparison just to validate that this method can allow you to discriminate between different strains or different polymorphs. Okay, so here we have a red profile. So if you have a low point, this is where a substitution to an alanine affects the incorporation into the fibril. And so we can see that here, this core is Viquivic. So this red strain is quite different from the gray strain, where the gray strain uses mostly R3, R4. In contrast, if you take two similar strains, you can nearly overlay the two. And so now what happens if you take one of these strain profiles that's derived from CBD and map them onto a structure that's been recently published? So this is what we get. So here, red positions are colored according to the importance for incorporation. So if you mutate this residue that's red, for example, this tyrosine, the strain, the sequence does not incorporate into the fibril. And so if we compare this to our in silico alanine scan, we get a remarkable correspondence. Okay, so we have this cluster here and here that are almost perfectly superimposable. And indeed, what we see is that these are the amyloid motifs. So these are the hydrophobic contacts engaging amyloid motifs that stabilize the fibril conformation. So now we can use the energetics from modeling and show functionally that these residues are also important for not only energetics of getting these structures, but also for the conformation of these things. Okay, so I want to sort of end on a couple of notes. So, you know, our model is that tau is actually a remarkable protein because it doesn't aggregate. How does this protein that does not aggregate change shape to these different forms? I don't think PTMs are a driver of this process, or at least not phosphorylation. We think that ligands or something that induces these local conformational changes around the amyloid motifs may be the driver to form these different forms of monomeric seeds that then can allow the formation of a diversity of different structures. And so a brief plug. So as Christoph had mentioned, I'm the recipient of one of these curePSP venture grants. And so we're really excited and thankful for the donors for this money. And so what we're interested in understanding is, what is the conformation of these small tau seeds that are derived from the CBD and PSP tauopathies? And so we have a structure of the CBD fibril. The PSP is not available yet. And so it'll be very interesting to see what is the relationship between the CBD monomer to these fibrils. What are the conformational changes that drive this process? And can we learn something about detecting these species early on? Okay, so I want to end on sort of a summary. And so, of course, we're interested in this really simple idea where local structure can control the exposure of amyloid motifs. And if you have a perturbation of this, whether it's mutations or ligand binding, we think this process leads to aggregation. And because tau has multiple of these elements, we think that the combinatorial exposure of these allows you to sort of build a taxonomy of different fibrillar structures that are now linked to different diseases. And so we're interested in understanding what are the local interactions that drive this process that prevent aggregation. We're also interested in how molecular chaperones mediate this conversion. Chaperones bind to this state, or do they bind to this state? And then finally, in collaboration with the Diamond Lab, we're interested in developing therapeutics and diagnostics that help us detect these species early on in disease, and things that are actually selective for the different conformations of tau that are all linked to these different tauopathies. Okay, so with that, I want to end on the acknowledgements. Of course, none of this work would be possible without all the funding and all the people that have contributed to this work. And so in my lab, Dailu Chen and Kane Dromboski, who's actually joined with Mark's lab, really developed the hairpin idea. Hilda Merbaha was really the pioneer behind all of the seeding monomer data. And then Chichang Hao in my lab as a staff scientist, he's really helped sort of characterize the in vitro production of the seed and all the biophysical characterization of it. Sophia Bali is a graduate student in my lab who's developed all of the molecular dynamics and the sort of comparison of the 3R, 4R. Vish Mulapudi, who's an undergrad, actually just graduated, who's developed the alanine scan in silico. And then with Jaime, we've been trying to understand what this alanine scan in cells means for our ability, for example, to model the different structures. Of course, collaborators, Mark Diamond, Milo and Levant for help with molecular dynamics. And with that, I'll take any questions. Thank you.
Let me start my video. Thank you so much, Lukasz. So we have a perfect timing. Thank you so much for presenting all this work. It's quite a tour de force. I really enjoyed that. We have a few questions now. I'll go to the first one. One second. So at one point you were showing the different parts of tau, and two people asked this question around Pick's disease and 3R aggregation, and related to what could facilitate 3R aggregation in Pick's disease based on what you presented.
So we don't, so for some of these fibrillar structures that have been published, there's additional density that's sort of, I guess, unexplained so far. Some of this may be PTMs, but the others don't seem to be covalent. And so it could be cofactors. You know, it could be things that are charged that are helping stabilize some conformation that helps expose the amyloid motif. But we don't really know. You know, I think heparin has been sort of commonly used in the field to induce aggregation. And I think from the structures, it's sort of obvious that the heparin-induced the fibrils are not the same as the pathological fibrils. And this indeed is also true for the alpha-synuclein structure. So in vitro produced fibrils are not the same, for example, as the MSA fibrils. And so what we do in vitro currently is, I think, reports on biophysical properties, but it doesn't really report on sort of disease. And this is something that we're really excited to try to figure out. But I think it's cofactors. It's just not clear exactly what they are. It could also be other proteins that bind to different ways, right? But right now, we don't know.
So another one, is there any possibility to detect the seed-competent monomer tau form in CSF of dementia patients and correlate their level with clinical trajectories?
Yeah, so the CSF is actually interesting because I think there's evidence that, at least in AD, the tau that's found in CSF is not full-length tau. And so we don't know if it's possible to detect this or activity in CSF. In other tauopathies, tau does not really increase in CSF. So we don't really know, but it's possible, for example, that we can detect this in exosomes or things that are secreted from the brain. And so this is still sort of ongoing for how one could actually detect this.
Thanks. Many years ago, there were suggestions of proallele isomerase genetic association with Alzheimer's disease. Any link to isomerization of P301? That's a specific question.
So we don't have a direct link. We don't know. This is a great question. Proallele isomerases definitely bind to tau, but I don't know if there's any direct evidence that they work on P301 to change this. One thing that we have done actually to sort of support that idea, sort of, I guess, in vitro, is we can introduce prolines that have a fluorine that can shift the equilibrium between cis and trans. And a trans-shifted proline induces aggregation, whereas a cis-trans shifted proline does not. And so we think the conformation matters, but we don't know if there's any isomerases that actually work on this specific site.
How stable are monomeric seed-competent species and phosphorylation sites during biochemical extraction process?
So how stable? So if you purify these seeds from tissues, you could boil the samples for an hour and you still retain 50% seeding. We have not assayed if phosphorylation changes the stability. That's actually a really great, it is possible it could change the stability. Although it's not evident that it could, but it's not evident that it necessarily has to, I guess.
Another one, could you explain more why you think that phosphorylation on tau does not drive tau accumulation?
So I can qualify our answer and say that phosphorylation is not important for keeping the structure of the seeding conformation. It's still possible that phosphorylation, for example, gives you some context that can stabilize it, but once you form that, it's not necessary to keep it there.
Could you comment a couple more? So this is great. Thank you for submitting all the questions. Can you comment on the similarity or differences between the MI and MS forms of tau versus A beta 40 and A beta 42 forms of amyloid beta monomers, which also have different propensity for aggregation?
So A beta is actually sort of, I think A beta is really aggregation prone, right? You can mix A beta and within hours you have oligomers and aggregates. In fact, you have to dissolve them with ammonium hydroxide to prevent aggregation. For tau, this is not true, right? Tau, normal tau, if you purify it, it will not aggregate. So I think there's a vast difference, I think a lot of it has to do with A beta sort of being able to form oligomers rapidly. And I think there's been some simulations that sort of suggest what these could be, but I'm not sure if there's direct experimental evidence for what those structures are. And so it's sort of hard to compare what that means for tau. I think the assembly of tau oligomers is probably somehow mediated through amyloid motifs. And so maybe the burial of these amyloid motifs prevents that. And so then exposure of that could promote the formation of oligomers. But we haven't done too much on oligomers. I mean, this is something that we're actually very interested in, because you have these sort of species that are intermediates sort of beyond monomer, and we really know very little about what they actually are. But they for sure seem to be important in disease.
Okay. I have while waiting for all to submit questions, I have a couple myself. When you showed the cross-linking experiment, you identify residues that are closer to each other for the seed-competent tau versus the normal tau. Are you using this kind of proximity map for potentially drug discovery, or is that, what type of data are you using to test particular compound that could prevent maybe aggregation of those specific forms?
Yeah, so one thing we're trying to do is sort of see if we can develop antibodies that are conformationally specific. I think the cross-linking is more of a tool, a discovery tool, because I think it's not really sensitive enough for diagnostics, for example, right? I mean, one could say, can you detect this conformation in a tissue using cross-linking mass spec? And in principle, I think it's possible, but I think it's not really sensitive enough. And so we're essentially using the cross-linking to guide how we think about structure, and then using more the insight from the structure to sort of infer how one could think about designing things that are specific for one conformation versus another conformation.
Yeah, and there's another question there that kind of addresses a bit of that controversy of where is the pathology present at a structural level. So the question is, do you think that most seeding events are carried by monomers, soluble oligomer, or insoluble oligomer? And what of these different forms of pathological tau do you think is more prone to support the spreading hypothesis? Big question there.
So in terms of spreading, the monomer, I think, is not very efficient at spreading. So I think you do need to form some sort of a small assembly to spread. I believe for entering a cell, at a minimum, you need a trimer, although there's some controversy whether tags can modify that. So we think the conformational changes in a monomer can drive aggregation, but they're probably not quite related to spreading. So you probably need larger things to actually spread and actually get sort of pathology, right? And so this is why this mouse experiment is very exciting. I mean, you can't do this experiment in humans, right? But in the mouse experiment, you don't see pathology until, six months. But yet at 4 weeks, we see evidence of seeding. And so this assay, this biosensor assay, is actually remarkable because you can really detect very small amounts of seeds that you wouldn't be able to detect on a Western, right? So we have a very high sensitivity of detecting sort of the very early stages of disease. And so if we could figure out when this happens in humans, we could look at people that are in their 30s or 40s and say, you have a seed. Mm-hmm. Let's catch it. So, but of course, if you can't see it in CSF, it'll be very difficult to do this, right?
Makes sense. Another related to the Cryo-EM kind of summary data that you presented, what drives the unique, in your understanding of the field, what drives the unique, the unique disease-specific structural conformation of tau recently shown by Cryo-EM? All the structure that are specific to disease, like what do you think drives this difference?
So one thing that sort of complicates all of this is you also have, contribution of other proteins, right? So in Alzheimer's disease, you have A beta and then you have tau, right? So technically Alzheimer's disease is a secondary tauopathy. And then, for other sort of tauopathies, they're mixed 3R and 4R, some are pure 4R, some are pure 3R, or I guess one is pure. So there's other sort of components to this. And I think it's probably something to do with interacting and stabilizing the conformation that can give you contacts to some other part of the protein that's consistent with the fibril. And so, of course, we don't know what the conversion from a monomer to an oligomer to a fibril is. We don't really know what the relationship between those two are. But what I can say is that, our induced sort of heparin monomer it makes sense in the context of the fibrils you get from heparin, right? So heparin binds to a part of tau, exposes it. We think that sort of the terminate can fold back, and then that, you know, Viquinc amyloid motif is exposed, and then you can self-assemble to form fibrils. And so that model for a monomer actually makes a lot of sense for the heparin induction model. And so I would expect that essentially it's ligands binding in a way that allow interactions that are consistent with the structure. And we're actually going to test this. We're actually in the process of trying to do some cryo-EM on samples that are MS sort of induced fibrils. And so we'll be able to compare how that structure is different from the heparin induced fibrils.
There's one more I want to ask before we wrap up, is that what in your mind is the origin of the formation of the seed competent monomeric tau? How does a monomer of tau become seed competent? What is the drive to?
I mean, maybe there's some aspect to, I mean, there's for sure an aspect of, you know, genetics that we don't really have a very good handle on, right? So there's risk factors for this. And I'm sure there's some effects of environment, you know, head trauma, for example, right? And so this is maybe how you get CTE. And so it's not really, I don't know. It's really hard to answer that question. It's a very, very broad question. If you know this answer, I think we could solve this problem. But I think until we understand how you get to these states, it'll be really hard to interrogate.
All right.
I will say that chaperones can be maybe a segue to this, right? So there's many molecular chaperones that can bind to these substrates and prevent their aggregation. And so maybe there's an aspect of sort of proteostasis where molecular chaperones maybe are depleted due to some other stress, and then you can form these structures. But again, I'm not sure how one necessarily gets to different conformations from, you know, genetics, I guess, right?
Yeah. All right, so we are on, it's 1 p.m. here, Eastern time, noon your time. So I want to thank you for your presentation, and we will share the recording of the presentation with the group for those that couldn't attend to the full talk. So thank you, Dr. Joachimiak, for showcasing your work. We're looking forward to seeing the new insight that you're producing with the venture grant and other source of funding that you have to support your great work. I also want to thank Jacqueline Zandrian, our VP of Events, for putting the webinar together. Thank you to the audience. We got a lot of great questions. Feel free to reach out via email for any other questions, and we will send an announcement email when we have the next webinar set up. In the meantime, you can visit our website, psp.org. Goodbye, everybody. Goodbye, Lukasz, and have a great weekend, everyone. Thank you.
Yeah. All right. Bye. Stay safe.
Bye. Yeah. Bye.